Lights, that's the way it goes. Okay, now what I'm going to do is I'm going to show a couple of pictures of cover crop, and then we're going to go to the new type of soil sample. This is the group for that, right?
Oh yeah.
Okay. We're pretty excited about the new type of soil sample that we're doing. But before that, I'm going to show this because Tom, he baited me. He says, "Well, Ray, how do I do some of these crops? How do I?" He's going to be an organic farmer. Right? You see that picture right there? That is a no-tiller. I want to show you, you know what that is? That's a 7-way mix. Cereal rye, vetch, crimson clover, rape, all that. He's a 1,200-acre farmer. He's never used covers before. Never used covers before. Not in a huge extent. In fact, when we started this, I said, Curtis, let's only do 10 or 15 acres. He did 850, 900 acres. I was stressed because he's never done it before.
Did you sort of let this get away then?
No, it was designed to go that high.
Oh, okay.
But he didn't realize, you know, it's interesting because what happened is he's a district board director. And he asked for help because he saw my presentation. He said, "Rick, can you help me?" I said, "Under one condition, you're going to North Dakota." He goes, "Well, you want me? North Dakota? Why would I send my farmers to North Dakota? Why would I send them to a different climatic zone, total different precept? Because I wanted him to see the power of diversity done on a large scale. If they could do it in North Dakota with only 16 inches, he could do it in there in North Carolina with 24 inches. The principles of ecology apply everywhere. Because the first thing people tell you, oh, it can't work here because it's too sandy, it's too wet, it's too clay. I hear that all the time. Oh, I'm sorry, ecology doesn't work here. We got heavy clay soil, we have sandy soil, blah, blah, blah. So here's what I did. We took him, we came back, we planted this, and he saw the biomass, and he freaked. I mean, he freaked literally. Okay, so we rolled the cover crop. Guess what we did? We pulled off the old cultipacker, the ones that break up the clods. So you had a paper roller, rolled it down. Look how high that covered up. That's why Ray Steyer's is 4.6% organic matter, ladies and gentlemen. He does that every year. Every spring, he's no-tilling right into that mix. You can no-till it standing, or you can roll it. Now here's where it got ugly for the first time. Using the no-till corn planter, we started having everything. We knew how to set the drill right. So I called Dave Brandt. He's a savant of no-till drills. Told him what's the drill number, boom, 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 boom. Do this adjustment. Bingo! It worked beautifully. Here was the key. Tom, when you plant cereal rye here, it is easier to kill it once it reaches the dough stage. When the green little pollens start falling off, you just roll it, and it is easy to kill. You get like a 95% kill rate, but it has to be at that stage. And you can't let it get too brown on you because then it becomes very lignite. And the drill can't cut through it. You don't want that. Here's the problem though with vegetable farmers. They can't wait an extra 2 or 3 weeks to let it go brown. Now here's the beauty about when it goes like that on a blanket. You know what it does? It has an incredible weed suppression. Cereal rye, pigweed hates cereal rye. Russian thistle hates cereal rye. It is an incredible weed suppressant. It's got some ileopathic effects and the smothering effect. It is beautiful. Look at that. You can almost do this without any herbicide. If you do put one herbicide, you put it right at the surface of a dead plant. Where I have a problem with Roundup is when you're putting Roundup on live plants because it gets sucked up into the kernel. But here, it's just has some revegetation, and the microbes will eat it. Most people don't realize if you have a healthy, good soil, Roundup can be denatured very quickly. It's got phosphorus and carbon changes, and the microbes can break it down. Now, I'm convinced, I wish that we would be using Roundup like drinking water, and that's why we're having issues with it. You only use it wisely, but no, we don't. We spray it very liberally. And personally, if you can do it without Roundup, even better. But you have a good crimper roller with a heavy water drum, a lot of water, make it heavy, you can probably get away without okay? Now, here's how we do some of our cover crop seed. That's cotton. We're spreading cover crop seed, and they're defoliating cotton. And look at this. Beautiful. Look at the corn come up out of that. Cotton pops right out of that. Beautiful. In fact, everywhere we do this with soybean, we get a 6 to 10 bushel increase. Why would we get a 6 to 10 bushel increase when we plant soybean into cereal rye? Larry, what do you think's going on? Producing nitrogen? It is. It sucks all the nitrogen first, and it causes the roots to nitrate more. And guess what else you're doing? You're lowering the soil temperature. I don't think people realize that skin, that upper part of that detritus, that soil temperature is critical. And it lowers the temperature, suppresses the weeds. That corn produced 180 bushel corn. Look at the weed protection. There's the corn. We got 3 bale cotton. You guys see any weeds there? This year, Dave Brand planted 14 acres of soybean in cereal rye. He had no weeds. Farmers came from all over the country, and they said, Dave, you're lying. He got mad and said, I'll give you $1,000 you can find spray tanks. So, no weeds. I saw that field. That soybean field produced 70 bushel. You see why the colors are so critical? Once we start understanding how to use them in the proper setting, they're very powerful. This is what no-till's been lacking, because what we do is we'll spray. And that's not what we want to do. That's not the direction we want to take it to. I'll talk more on this other one. What's my other session, by the way? What's the title? And we'll talk about the soil session. Faster. And I'll talk some more cover crops there too. Let's go to the fertility one, because I don't want you guys to miss out on it. Do they have any schedule for any topics tomorrow? We talked about it. You see the schedule? Am I scheduled for any?
Yeah, we've got you scheduled, Ray, and if you've got to get on the airplane, then your understudy will handle it for you.
Yeah, I don't have to leave until noon.
Okay.
Okay? But let me show you, let me bring this up. I'm going to tell you about the new soil sample. Now I'm going to present some blasphemy to some of you, because it's going to be a different way of changing. I could tell when I was telling Mark about some of this stuff, you can see his facial expression. He goes, oh, don't do this to me. And let's go here. Down the bottom here. No, I like the.
No, I do too.
I like the work. Okay, thank you, sir. You knew I needed water for my voice. Okay. This soil sample are based on green chemistry, ladies and gentlemen. Say it again, green chemistry. The Malik and the Bray use very caustic materials to knock things off the exchange end, doesn't it not, Mark?
It does.
Okay. That's not what nature sees. It does not see those kind of chemicals. It sees plant exudates. In case some people don't know what plant exudate is, it's what those plants leak. Each plant has its own chemical signature. We're finding out through new science that plants communicate to each other biochemistry. They'll actually communicate and send biomolecules. When they have a certain pest plant food, they will send this soup into their biomolecule, and they'll also send it through the mycorrhizae. They'll communicate to each other. And they say, hey, we're getting bitten, so we'll send off these alkaloids. How many of you guys had coffee this morning? Every one of you drank insecticide. Coffee is a natural insecticide. Nicotine is an insecticide. Aspirin is an insecticide. So when people get a little perspective, you got a perspective about the word insecticide. Okay? Some of it can be the problem with insecticides we create as man always have these side effects. But nevertheless, let's go here. This is where you're going to send, we send soil samples to now, Dr. Rick Haney. Interesting is that just gave him $100,000 because we're going to support this soil sample because we love the concept and we're getting better answers. See, when it came to no-till in pastures, we would see this incredible, excellent no-till field, and you get the University Services, and it's there, and you put more nitrogen. And we're running a SPAD meter, and we have this, and it says, no way, it doesn't need any more nitrogen. The model's wrong. Ladies and gentlemen, our soil samples at the university were based on till systems. They were not based on systems that are in dynamic equilibrium. Pastures and no-till systems is dynamic equilibrium system. That means they're at a certain equilibrium, but they're always dynamic. Wetting, drying, cycling. You cannot compare these two soils. You can't compare them. In fact, yeah, you cannot compare these two soils. They're total different animals now. Nutrient management is totally different from them. The inorganic soluble state model works very well. The oldest model from the University we get for soil samples works fine for this. But it does not work very well for no-till and pastures. Now, the Rick Haney test will also work in conventional, but you cannot go till the soil and then go take the soil sample because it's going to be totally wrong. What this sample is going to measure is the labile carbon. What does the word labile carbon mean? It's the active carbon. It's like the food that's easily digestible. There's passive carbon, which is the house, the construct. When I say total carbon, organic matter, it's like the trellises and the beams. That's the construct. That's the house. Okay? The labile carbon is the food. It's the easily accessible food. And that thing, that pool, is 80 times smaller than the total carbon. So if I know how much food, and I know how much the bugs are going to mineralize, how much they're going to convert from the organic to the inorganic, how much nitrogen and phosphorus they're going to make available. Okay, everybody with me? Now, who makes nitrogen and phosphorus available in the soil? Hey, by the way, if you come to my class, you're going to be called none of us all have the answer. Come on, man, what do you think? Who makes phosphorus in the soil? Come on, guys, help them out.
Fungi.
Speak out.
Fungi.
Bacteria. The bacteria guys. When we put phosphorus in the soil, what happens to it? Right? It binds up, doesn't it? So how do we bind it? Does it happen by itself? No, it doesn't. It's these organisms excrete these phosphatases, these powerful enzymes, and they can change the pH around them and make it soluble. Also, roots from plants will leak citric acids. These are exudates can be acidic, and they make these nutrients soluble in the soil. It's biologically done. Okay? So we know this now. And this is the tools we're going to be using because the ones that we've been using have been inadequate. You see these little carbons right there? This is the complexity of organic matter. This are these molecules that these microorganisms just strip and eat as food substrate. Okay? This soil sample has the ability to sample that. Oops, wrong PowerPoint. Boy, I only put a couple on that one. Oh, here it is. Apologize. So, any questions so far about the test? What it's going to measure? Now's your chance because I'm going to tell you, and I'm going to go over one of them. I'll show you. This is Dr. Haney's test. Let's start right here. Okay, now this is ARS. This is not raised voodoo science, okay? Because some of you that came from a very strong agronomic, all of us came from a very strong inorganic chemistry. How many of you got a degree in agronomy here? Went to super agronomy? Anybody? How about soil science? Really? Okay, well, good. In the sense, you don't have too many paradigms. It's actually, I'm an agronomist, so I know I'm tough on this to be able to have paradigms. I walk away soil chemistry stuff. The soil is driven by biochemistry that runs inorganic chemistry. Okay? Now, this research papers have not, this test has not been any peer reviewed research. Okay? Now, this new soil test not only measures the NP&K, soil organic NP, microbial activity, water extractable carbon. Now look at the key word, water extractable carbon. Okay? Carbon nitrogen balance. Okay? Measures activity. Because it's these guys that drive the system. Okay? If we know how much food is in the system, we know how much activity we're going to get. They're the drivers. Now, when we're all taught soil science, we would do this little schematic, and it would be small, but we would focus on all these little things, but really the focus should be on the microbes. Let me give you an example. In the early turn of the century, there was a scientist, he was Russian. They've known for a while that soils go through these cycles: wet, dry, okay? Now, let's, for a split second, let's all imagine that we're microbes, okay? Life's tough. We had a severe drought. Have you guys ever had drought? When was your last drought?
Last year.
Last year, you guys suffered real too. Okay, let's say drought was a little tough. Jack dies, Tom dies, we're all microbes, life's tough. But some of you other guys survive, rain comes back, all of a sudden, there's pieces of Tom, pieces of Jack, pieces of organic matter. All of a sudden, the activity starts going back, multiply, there's food in the system. The higher the fertility the soil has, the higher of CO2 respiration that first day. It's called the CO2 respiration on the first day. That's why we call it that CO2 release. Because see, soils do this. They go through moist cycles, dry cycles. Moist, dry, moist, dry. In that release, there's a huge release of CO2. Now these are three soils. The College Station was the least fertile. Lubbock was more fertile. Amarillo was more fertile. Why is this one more fertile? More food substrates, more carbon, more huge microbial biomass. We now have science saying that these dead parts of Tom and Jack make up maybe anywhere from 30 to 40% of the organic matter. Dead microbial bodies laying everywhere. Very cool. So that's a food substrate, okay? See, in the soil, everybody eats everywhere. There's no way to use that. Okay? Now, here's the instrument that we're using. These are about $1,400 to about $1,600. We just bought one before the government went broke. So I want to work with producers. What it does is got a paddle, and it's got a gel substrate on it. And that gel is very sensitive to CO2. It's very well calibrated. This has been gone through a lot of research. Okay, so what you do is you go collect your soil and you put it in this glass jar. You put it in this container. And you only disturb that soil in that part. So keep in mind, say I'm going to be a cotton grower or I'm a corn grower. When does corn need nitrogen the most, folks? At what time?
Grain fill.
About the V8 stage, about 8-leaf, 10-leaf stage, right, Mark?
Okay, sure.
Then it goes to R1. The demand of nitrogen is about 200-fold. I mean, it wants nitrogen at that time. At the early stages, it doesn't need as much, but at that time, it really wants it. So, if I can't send it to the lab, and I want a quick response, I want to know right away, I will go over there with my little paddle. You don't have to buy the digital paddle. It's got a color key. Then what you do is you tie the jar and you keep it for 24 hours. Closed. Okay? Close the jar, and then the paddle will turn color. Now, if it turns yellow, you got more than 140, 180 units in your soil. If it's that color, you have very little activity and very little mineralization going on. Very cool tool. so I can get quick field response right away. Okay? But the more accurate one is we take it to the lab. Now, keep in mind, this organic pool is about, so 2% of soil organic matter, you'll have 12,000 parts per million of carbon. Here's a number you want to keep in your head. So when you go to a cocktail party, you want to impress somebody. You know organic matter has 58% carbon on the average? Yeah, that's a way to pick up dates. Yeah, or pick up a rash than anything else, right? Now, we know when we did percent, there's a great correlation between soil organic matter carbon. Now look at this, right here, in water extractable carbon. Look at this vein. Look at the organic matter 6.4. Here, Wyoming have 5.96. Look at the water extractable carbon, 4.6. Yeah, pretty direct correlation. Pretty close. Look at the soil microbial activity. Look at the vein, more organic matter, more activity. Pretty close. Okay? Now, here's another thing we're finding out. This was never taught to me in college. See, I was taught in school that plants take up nitrogen in what two forms? Ammonium and nitrogen. But that was pounded in us, right? But they never told me that plants can take up organic and, say it again, plants can take up organic and through the mycorrhizal fungus, and they suck up amino acids. Amino acids, guess what it has in there? A nitrogen in there. So, think about this. Our forests and our prairies are 95% of our nitrogen in forests and prairies are in the organic form. So if they're in the organic form, Mike, why aren't they nitrogen deficient? Because the plants have the ability to access it. Here's another thing. If plants can take up organic molecules, you can take herbicides out. They can take up organic molecules. So what I'm saying is to you, why is this important? It is important. Because you need to understand where nitrogen is coming from. It's coming from the leakage of amino acids, from certain, like cover crops, nitrogen fixation, and there's another source that we never account for. Anytime a nematode, I call them the hyenas and cheetahs, eat bacteria like the gazelles, they multiply billions. You can have billions of them in weeks, trillions, bazillions of them. When they come and nematode comes and eats one, bacteria have a carbon nitrogen ratio of 8 to 1, 10 to 1. Protozoa have a 30 to 1. When that nematode eats it, excess nitrogen is released in the ammonium form. So, just by predation, they can get anywhere from 16 pounds to 116 pounds of man through predation in a year. So here's what I'm getting at. What is the most limiting factor in the soil? What would you guys say? I was going to ask you what is the limiting nutrient in the soil. What would you say? Jim? I would say 2. Nitrogen. Jim said everything nitrogen. 3 years ago, you would ask me that, I would say the same thing. I say it's carbon. You can't separate the carbon and the phosphorus in the nitrogen cycle from it. See, the microbes want carbon, and they use nitrogen as an energy substrate. They're together. So, if I build the carbon in the soil, I'm going to get more NP. Okay? Yes, ma'am?
We've been taught a lot that if you overload carbon, you can tie up that cycle and you can nitrogen starve.
Yes, you can. You see, under the situation, under tills, what have you done? You tilled it under, and you increased the surface area, and you stole all the nitrogen out of the system. Yes, because the numbers of microbes are growing so quickly that they're looking for any nitrogen in the system.
So as long as we leave it on the surface.
When you leave the surface, it doesn't happen. Because it's happening slowly by the grinders, mixers, and shakers. Now that residue on the surface is 20% of the soil's energy. 20% of the soil's energy is that residue. But most of it's coming from those exudates. It's more powerful than we've imagined. So we want to be able to get into those pools those intermittent pools of N, and the water soluble. Everything I say is water soluble. There's the key word. Water extractable carbon. Water extractable nitrogen. See how cool this is? Now this makes sense because this is what the root sees. It doesn't see the caustic chemicals of the malic and the bray. Now, what we want to do is, then also this test will tell you the balance between carbon and nitrogen. It'll tell you how much. So we know that if we have too much carbon in the soil system, like you said, we're not going to get mineralization to happen. So Rick's test has an ability to check the carbon and nitrogen balance ratio in the water solution where the microbes are at. Okay? That's very cool. I'll show you. Now, here's the parameters it's going to check. It's going to check extractable carbon, the percent MAC. Percent MAC is the microbial extractable carbon, the process of food, extractable nitrogen, inorganic NMP, and organic carbon and nitrogen, and organic NMP, and the Savita one day. Those all those parameters, and you're going to get a soil health calculation. If that calculation is less than 7, I'm going to write this down. If it's less than 7, you have problems with your soil. You want it to be 7 and above. Okay? Now, this is a full day course. We're going to have to come back and teach. What the whole goal is we're going to be able to teach our people how. And then we're going to have Dr. Rick Haney give a webinar. And when that comes out, we'll see, and then we'll teach you how you can read the soil. I'll show you in a little while. Okay, so when you send your soils, you're going to do it like a regular soil sample at the university. 6-inch depth, guys. Not 4, not 8, not 9. You're going to take it 6 inches like a regular soil sample at the university. Okay? Same thing. Now, you're going to say, when do I sample? Well, what's your goal? What's your goal? Do you want to do a benchmark? Or do you want to know how much nutrients you need to put out of food? That's when you take your soil sample. Okay? I personally would do a benchmark first. So where I'm at. Then, if I got corn, if I got crops out there that are high cash, I'm going to go take it into a container. Now, remember I showed you that little container? That's called the bill. Dr. Brenton and Haney came up with the same test, okay? Now, the one I'm talking to you about right now is that you're going to take it and send it physically to Temple, Texas, okay? Now, it's going to be free for a little short while. If you guys, because you guys were good little students, you can buy yourself a sample and you get it on the say it's Ray Archuleta, and you'll get a free sample, okay? And then it's going to be $50 for that $20. Now, some of you are going to say, $50. You know what I said? If you're not willing to spend $50, I don't have time to visit. I have one farmer who saved 60 grand by using the soil sample. You get paid about $50? Really? Okay? Now, you're going to send it in, and he's going to run it through all this sophisticated equipment. Okay? And you're going to get everybody have Excel at home. Everybody has a spreadsheet at home. Is there anybody that doesn't use a computer or does not have Excel? Okay. You go on Google and type Open Office. You get a free Excel spreadsheet. It's called Open Office. And then, because he's going to email it, here's what we're doing. We're in the process right now. When we get the website open, you're going to be able to put your name, your address, and all of it. The information will go soon into his Access database. And then your soil sample will come back with the results. But right now, we're going to get an Excel spreadsheet coming back. I'll show you for a while, okay? So, this is the way it's going to come back. It's going to go back in an Excel file. And this is the way it's going to look. Okay, it's pretty simple to read. I know it's hard to read from there, but here's what you're going to get. You're going to ID your soil samples. Sample 1, sample 2. You name it, okay? You put it on your thing and name Now, you're gonna do, so you're gonna say, how do I go in the field with soil sample? Say, if you have a sandy soil, and you have a clay soil, say you have a 40-acre field, 20 acres is really heavy clay, 20 acres is very sandy. Isolate them, treat them as separate fields. And the more soil samples, and take a 5-gallon bucket with you, and have an auger, and take as many 60 samples as you can of that particular spot. The more you do that, and you mix in the 5-gallon bucket, and you set a couple of pounds of soil to it, the more you do that, the more accurate the test is, okay? You guys all know how to do soil samples. Is there anybody that has not done soil samples before? Pretty sure everybody's pretty comfortable. Okay, so it's going to come back, and it's going to say the N, the PTO5, the potassium, and the nutrient value. It's got a little button here, calculate. I'll show you how that works in a little while. And you're going to put in the crop, and then it's going to tell you how much nitrogen you're going to need for the crop. Then it's going to tell you the reading of sulfida, the organic N, organic nitrogen, the organic carbon-nitrogen ratio, and the soil health calculation. And guess what else Rick did? It will tell you what percent your mix should be, the composite of your mix cover crop mix should be to address the issue. If you have very low, if you have a lot of nitrogen, like this one did, you're going to only have to put 10% legumes and 90% grass. Pretty cool. So we're using that as kind of a foundation as to how to set up our mixes. So if I want to know how to design my mix, I first take the soil sample. That's why when people say, well, Rick, what do I use for cover crop? I really rarely like to give recommendations about cover crops. Unless I know what you need to be, know what your goal is and what you're going to do. Now look at this one, for example. I want you to notice the percent, when you see this value, if you guys probably want to write this down. When you see the sulfida, you should be in the range. If you have 30 below your sulfida one day, that's low. If you start reaching the 50s, 60s, 70s, that's where your range you want to be. That's the respiration you're releasing. Now, can you guys see that little red triangle in the corner right there? You see that? When you put your cursor right over it, it gives you an explanation of each of the columns. And then when you send this soil sample, Rick has a complete write-up explaining all the details of each parameter. So if you don't remember everything I taught you today, that's okay. He's going to send you a written explanation of each parameter. Pretty simple. Okay? Now, notice this one. I'm going to show you two. I'm going to show you a low number. It's still not too bad. The soils are pretty good. Let's look at the top one first. Look at the percent organic in water. That's high. If you have it in the 40s like that, that's pretty darn good. But look at the balance of carbon-nitrogen ratio. Now, we like to see that number 20 and below, carbon-nitrogen ratio. If you have 20 and above, you're going to have tie-up. So that's the key number. We like it 20 and below. Okay? Now, notice here, the soil health calculation is 18. Now look at this one, this is the lowest one. This soil calculation has 22. So why is this one higher? Look at the carbon-nitrogen ratio. It's 10, which is excellent. Excellent cycling, lots of carbon, food. When you start having organic carbon, you have more than 400 and above, or 350 and above, you got a lot of food. This is the food, the organic carbon and water solution. This is the food, and this is the energy substrate. Both the organic and both are needed. And the ratio of it is important. I want that ratio to be 20 and below. Okay, everybody with me? So notice the number of the drop here. This is why, look at the soil health scorecard here. Look at the number here. Don't read these numbers isolated. They're all connected. 22, he says put 100% grass. You got a soil health score of 22. You got your carbon-nitrogen ratio is 10.3. That means the microbes are cycling very quickly. Got a lot of nitrogen, got a lot of food, great respiration. Now look at this one, the 7. Fraction of 7. Look at this one. Carbon nitrogen ratio is higher, not that much nitrogen, less food. Do you see that? So, focus on the food, carbon nitrogen ratio, and then it'll tell you how much in is going to be mineralized. That will give you that calculation. Now, I'm surprised some of you haven't said anything about, but Ray, how about pH? Has that crossed your mind? Okay, let me tell you about pH. As an agronomist, when we were at school, pH was almost like a god. The god of pH. Well, I'm here to tell you, pH is important, but it's not the big game changer as we used to think. Why do I say that? Have you folks heard about the concept called niche construction? We now know that the biology shifts the physical and the chemical. Here's what we're saying. For many years, ecologists thought that when organisms, Darwin's theory was that the habitat, that the environment, animals adapt to the environment. But he got it partially right. We now realize the organisms change the environment. We modify our environment. Earthworms modify the environment. Earthworms can change the pH of the soil, ladies and gentlemen, through their activities. Bacteria, all these organisms, all together working in synergy, creating those aggregates, create the construct. When they build that aggregate, they help modify the pH. They build things, and they put things in proper equilibrium. And then you create these miniature swimming pools inside these aggregates, and you help modify and buffer huge extreme changes in chemistry, one way or the other. So here's what I'm getting at. Within one aggregate, they're close to each other in the soil, there could be a whole pH difference. Whole unit. Microbial, we change the whole pH unit right around them by excreting these powerful enzymes. They can lower the pH. So can bacteria. Is pH important? I'm not saying it's important. I said take it in context. Because if you don't interpret it within carbon context, you're going to make mistakes. So here's what I'm going at. Ray soil, do you know what its pH is? 5.8. 5.8. Did quite well. Now, when I put calcium out there, I put calcium, in, especially in areas like this, and some humid areas, it's because they leak heavy rainfall. So I put calcium in the soil to feed the plant. It's not always necessary to modify pH. And then also, like Mark will tell you, to balance the magnesium ratio, you need calcium. Besides, also, calcium is a very powerful element that soil needs. In fact, roots will not grow without calcium not being present in the soil ecosystem. 5 minutes? Okay? So very critical. Now, what else can I tell you about the soil sample? Okay? We're going to get into a point that when we start rolling this whole thing out, there are 3-4 labs opening right now. Rainwater, and we need to write this down. The lab that's going to be doing this area probably, there's going to be 2 labs. There's going to be actually 4 labs that are going to be coming up. Noble is going to set up a lab. Ray Ward lab in Nebraska. You get to Google that. That's going to be a lab that's going to be doing this. Noble's going to start doing there. And Woodland lab in Maine also. And Woodland's going to be pretty elaborate because it'll do the other samples too and give you one total sample. I mean, it'll tell you much more detail than Rick's going to do. Rick eventually wants this to go into the private sector. He's a researcher. It's intended to go into the private sector. Okay? NRCS is supporting this as a tool because it's been one of the better tools we've done when I would do nutrient management and conventional no-till, especially on no-till, especially in dairies. This is the tool I'm going to be using. I'll use the university soil sample just to find out how much total organic matter, but this is the soil sample I'm going to be using. Why? One of the things we should know, how many of you guys write nutrient management plans? Anybody? one of the key things, like, here's what I watch. If I start seeing more than 200 pounds of human soil, I tell the guy, stop putting manure. Because you shut down the mycorrhizae. When you start reaching 150 to 100 parts per million, you shut down mycorrhizae. Why should I care if I shut down mycorrhizae? You're going to start having phosphorus deficiencies, and you're going to start having zinc deficiencies. Phosphorus is made available by the mycorrhizae. Zinc, all those trace minerals. You shut down the mycorrhizae, and then you're also going to make your soil drought resilient. It is the mycorrhizae that help us get through the drought. They have the ability to extract water in the drought. Luckily, 80% of our plants are mycorrhizal. Brassicas are not. Tillage radishes are not. So, we got to keep that in mind. So when you're doing this, so the soil sample we're going to be using, and there's the guys, there's one of our NRCS, there's the address. Go ahead and write that. We're done. Open up some questions while you guys are writing the address. Yes, ma'am. The Cornell test, I like the Cornell test. It tells you physical, chemical, biological. The only thing I don't know about the Cornell test, they won't tell me how they do the biological. So that makes me leery. I'm comfortable with RIPS. I'm not opposed to Cornell. I will use Cornell problem, no problem with it. But I'm telling you folks, you need to start realizing, remember when you look at soils, they have these three readings, chemical, physical, and biological, and they make them equal size? That's erroneous. The biologist is a lot bigger than the physical, than the chemical. That's the correct way. They modify the environment. Let me give you an example. I didn't finish my thought. Niche construction. We'll wrap it up. Niche construction. Bacteria, all these organisms change the environment of the soil. Beavers, they modify the rivers. The plants, they bring in the climate. They modify the climate. It's a two-way arrow, folks. Only life can beget life. It's biology. We weren't taught that. We kept the biology kind of good because it was so complex, it's unpredictable, and we wanted to just go. So when we went to school, we took graduate level soil physics, soil chemistry, through kinds of equations, and like Mike, right now, that's what I did in school. Really, I mean, but now, if I get the biology right, and then do what Mark likes to do too, is then bring in the chemistry, but you won't have to be bringing all the chemistry over and over and over and over again. I'll leave you with this last thought. And this is for you, Mark.
Okay.
All of us, really. I had a producer that really got caught up in trying to balance all the trace minerals and all the nutrients and getting into really, I mean, and he sat there and he's an organic farmer and he's a good farmer. I said, it costs a lot of money to play God, doesn't it? He looks, he got real quiet. He says, it costs you a lot of money to do this, isn't it? He goes, yeah. I said, why aren't you letting the biology help you? See, they will make the system in balance. They will keep it in balance. We can't afford to be just dumping line and all those input. Now, keep in mind, you're using petroleum, ancient petroleum, to move the line to the field. You line when you feel you necessarily have to line. When it starts getting to a pH of 5.5, I get worried. For my rhizobia bacteria need pHs of 5.5 and above. Then I'll start getting worried about pH. Okay? So, keep pH in context to carbon and to biology. You'll be fine. Anyway, one more question. You guys? Yes? Okay, two questions.
Have you tried to adapt this to agriculture?
Oh, yes. We're taking it all over. Rick Haney and all of us are building soil database. That's why NRCS gave him $100,000. We're going to build a massive database from all over the country. And we're doing that right now. All kinds of crops, pastures, everything. We're collecting. Now here's the kicker. If you're going to get a free soil sample, you have to share your yield with us. We want to know how you're doing. You're not going to be sharing with anybody.
There's like in the orchard,
We've been doing it in pecan orchards. See, when we were taught in pecan orchards not to plant covers in between because they steal water and nutrients from each other, we're finding that's not true. They're collaborating with mycorrhizae and they're sharing resources. Now you create a better habitat for predators so they can eat the aphids and the pests. But if you go in there and till every year and destroy the soil in between, now you're going to have to bring in the nutrients. Now you're going to have more disease problems. You're going to have more pests. No. Bring in multi-species cover crops, lagoons, pollinators, and everything. And then collaborate with the system.
What's your feeling with gypsum on the soil?
I don't have a problem with gypsum. We got to remember, it is a salt. Be careful with salts. In biblical times, the way you got rid of your neighbors is you salt the land. Nitrate is a salt. It's an elemental salt. We have research showing that when you have more than 80 units of N of some of these high salt fertilizers, especially in no-till, we knock the enzymatic activity and we lower our yields. I'm looking for fertilizers or low salt fertilizers. Google low salt fertilizers. Don't use high salt fertilizers. Compost, manures, covers, grow the biology, do as much of that as possible. Be careful with your salts. Do you notice how you say don't use them? I don't want people thinking I'm Mr. Organic. It's not what this is. It's beyond organic. So I want you to be careful with your tool. It's a tool. And let me tell you, Dave Brandt this year, he set some field, parts of the field. One part of the field, low nitrogen, 165 bushel. One part of the field, he put 50 units of N, 265 bushel. But in another field, where we had 90-day corn, produced 180 with no fertilizer. So there's all kinds of variables. So we're not real quick to say don't use chemical fertilizer, just use it wisely.
That'd be calcium, right?
Huh?
It's calcium, right?
Is it calcium?
Gypsum.
Yeah. I want the calcium. Calcium is incredibly important in uptake for plants.
I was just going to ask, in some farming circles, it's kind of seen as like a snake oil, but soil inoculants, you guys had a lot of chance to look at.
Let me tell you, the research is mixed. Some of them actually do work. But here, let me give you a perspective. That's why you never hear me as an NRCS employee promote any inoculants to a farm. Because if you bring in this one species and you bring it into the soil, now think about it. That soil is like the bad side of Chicago, and these guys are rough, tough guys. And you bring in this little genetic thing you've made, or you think you isolated, you put it in there, and it's going to get its thing kicked. So it doesn't even manifest itself. But some guys are catching on and saying, no, we'll put 100,000 great type of bacteria and put them in there. But here's my concern. Now, I spend all this money, this oil, this energy, this resource, and ship to you, and I try to make you addicted to my product. I could have just got you to make a multi-species cover crop and done the same thing. Okay? But here's the thing. Do I have a problem? No. I've seen some of them work very, very well, especially with microbial spores. If you have a very, very degraded system, I can see the purpose of it. If you have compost teas, you know what, everybody know what compost tea is? Okay, is when you get compost and you put molasses and kelp and nitrogen source, you aerate it, you've got this incredible material, you've got the microbes. Research from Odell Institute showed they do increase yields. But again, you're liable to only use them for a short period of time, because if you go out there and you're still killing and destroying the soil system, and you keep bringing all these inoculants in there, you're still burning the house down. It's like this rancher told me, he says, Ray, I'm never going to start disking because you know why? I get to prime my soil. I mix the cover, I prime it, and I get a yield response. Yes, you do. You burn the house down to warm up a hot dog. Just understand tillage is not your friend. But if you have to till your organic, it's really control weeds, then you need to bring in these other things you put in the system. Compost, seeds, very cool. Use them. But what I'm finding out with young people, they're not using enough rolling and enough no-till. They can use a lot more. And so that's not kind of, and the no-tillers, they do too much fertilizer, they do too much herbicide. They can use much more covers and be way ahead. See where this is going? So me as a holistic manager, holistic thinker, I never want you to be addicted to anything. I want you to be in a position to say, my soil's so healthy, I will use it if I want to. That's a cool position to be put in. Yes, ma'am.
We have a lot of corn and beans in our county.
Yep.
And we struggle getting cover on.
Yep.
We have something called corporate farming happening in our area where we have single farms taking over large areas of land, and they literally don't have time because they're chiseling up until the snow flies.
Yes.
But even if they did not chisel, they're harvesting till December, and they don't have time to get a cover in. But we're trying to talk these guys into aerial seeding cover or putting high boys and all this other things on, but it's a big barrier for us. We're really struggling with it.
Well, let me show you something real quick here, because I think you'll really appreciate this. Because, you know, I hear that all the time. I mean, I wish I could give $1 for every time I hear that. It's like, we don't have time. No, you don't understand the importance of this, because let me tell you,
Because if you really understood the importance, have you seen these high boy sprayers now?
Sure.
You know what they are now mixed with? Cover crop sprayers.
Great.
They have these tubes, and they are putting cover crops on standing corn. Now, why would a big corn and soybean guy spend $200,000 on a piece of machinery just to put cover crop seed and use for a sprayer?
Reduce the amount of synthetic fertilizers they use.
Because if I can get my cover that high,
Oh yeah, before you take it.
I can get 6 weeks. My cover crop's this high now. I'm going to show you a picture as soon as it comes up. But what's so neat about this, ladies and gentlemen, think about it. We have these big, huge rigs now that are going and they're spreading cover crop seed, and they're shooting the cover crop seed at 100 miles an hour down these tubes, and they spread the seed. Now, as they're harvesting the cover crops that high, what have I done?